All right, everyone. I want to welcome everybody to our second Polyploid webinar here, or as I believe Brittany Sutherland called it, our Polyploidy in Place, which is as good a name as any for this series. Um, Today we have, for our first speaker today, we have Dr. Tony Heitkam from the Dresden University of Technology. She has been there since 2019 as a lead scientist and also pursued her PhD there. She's recently started a new project on studying the identical genomes but different properties. Saffron as a model for epigenetics of quality traits and environmental adaptation. And today she's going to tell us a little bit, I'm sure, a little bit about the work that goes into that grant and the research they're doing right now on the genetic origin of the cultivated triploid saffron crocus, which I'm really excited to hear about. And I want to thank Tony for taking some time from her evening today to tell us about her research on these interesting plants. With that, I'll let you take it over.
Yes, thank you for the nice introduction. It's actually not so much about the new grant because the project will just start, so the start has been postponed, as you can imagine. So a bit more about the research going there. So yes, I will tell you a bit about saffron crocus. So, but just at first, because you know Dresden and we are spread globally, I just give you one very quick glimpse of Dresden. So that's where I am. So very old European city. But because it's not about Dresden, I want you to imagine some crocus flowers growing and thinking about a season where crocus might grow. And actually you could think about two seasons because some crocus species flower in spring and some in autumn. And I will tell you a bit about the autumn flowering crocus sativus, which is the saffron crocus. So on this lawn, there is no crocus flowering. This has been photoshopped. I think it's obvious. So saffron crocus actually is a source of saffron. And saffron is a spice you can buy in the supermarket, probably for a very high price. And saffron, the source of saffron are the stigmas of the flower. You can see here quite nicely. And can you see my pen? Yes, that's good. So there is a stigmas of saffron crocus, and they harbor about 150 different chemical compounds that make the unique color, the taste, and the aroma of crocus. And it is actually around for quite some time, so about 4,000 years, and we know that because some frescoes have been unearthed about 50 years ago, and they have been dated quite well, and they come from the Greek island Santorini, which has been devastated by a volcano explosion about 3,700 years ago. But these frescoes were very well maintained, and you can actually see here in the background here some crocus flowers, and here the harvest. So we know that saffron cultivation is going on for already quite some time, at least in Greece. And I want to take the opportunity to introduce you also to this photo series from the Iranian Students' News Agency, because you can see very nicely what goes into the saffron spice. So you can see where saffron grows in rural areas, on marginal soils. You can see this here. You can also see that the whole village gathers to help in the harvest. And you can also see while it's left for drying, it's not really left, it has to be guarded because it's so valuable. So a lot of effort goes into the spice, which is the reason that it's so very expensive. So you need 150,000 flowers for a kilogram of saffron, which will give you about 10,000 euros in the end. So it's one of the most valuable spices in the world, if not the most valuable spice there. And this is also the reason why it's often falsified. So you should never buy this, the ground saffron, because it will contain everything, but probably not saffron. And even if you go with the stigmas, you can actually have safflowers that are colored, or even maize cut to pieces and colored. So you have to be careful. And you can see here, there are many research projects going on to check where it comes from for its authenticity and so on. So there's a lot of research out there on saffron, not just the genetics, what we are doing. So saffron production today, most of it comes from Iran, which is here. It's over 90% of saffron's production. But there are also some other sites like the Kashmir here, which is where there's a lot, Spain, there's a lot, and we also hear about Greece today. But there's also some in the US here, and so on. But if you, so I can see that actually right now, but I suppose that you can, because there are some videos like on top of that. But I suppose you can. So if you take samples from the different sites where saffron is grown and just check for genetic differences. So you can see here some molecular markers, like here in the upper side, here is saffron, like here is saffron, and like here, all of that is saffron. It looks all the same. So you cannot really trace any genetic differences inside this. So by that, you probably know, because we're the Polyploidy webinar, and I already told you that it's a triploid. So it's a triploid, and you can sort the chromosomes into triplets. And to give you some idea about the genome size, the triploid genome size is over 10 gigabases, and triploid has 24, saffron has 24 chromosomes. And obviously the segregation is disturbed and it's sterile. So what we know what happened is that from the pool of 100 species in the genus Crocus, triploidization happened about probably 4,000 years ago, and that saffron crocus with a triploid chromosome set emerged from that. But we don't really know what went to that. And then we know that it was distributed and grown all over the world by humans. So saffron is one clonal line spread by humans, which with very high genetic identity, and probably it probably has just emerged once there. So that's the starting point here. It's, as you can imagine, exclusively vegetatively propagated. So you need to wait quite a while to get offspring in a way. So you have to wait. So it comes from corms here. You have to wait a few vegetation periods to go to here, to the daughter corms. So you need to wait a few years to basically have something to give to someone else, or to grow the saffron on a field. So it takes a while, and it's inaccessible for breeding, obviously. So yeah, we had that. So the questions that we wanted to answer, or that we are trying to answer, is how and when did saffron crocus arise? What were the parental species? And do the parents belong to the same species? And in the end, maybe make saffron accessible for breeding. And we didn't really start here with around 100 species, but there has been some work before that, so we could already narrow that down. So there has been quite a lot of phylogenetics work going on, and we know already that it's very, very closely related to this species here, Crocus catwhitianus, the diploid. So this is already quite accepted as one possible parent. So we thought it could be either an autotriploid by a triploidization with one parent, Crocus catwhitianus, or well, an allotriploid with genome components from a different plant as well. So there have been some candidates here. And so what we did was quite straightforward. We did low coverage skim sequencing of the Crocus sativus and an analysis of the reads. And because we do a lot of cytogenetics, we went directly, we used the reads to identify chromosomal landmarks, to have a closer look at the chromosomes, and to compare the chromosomes to the possible progenitors. So we went directly for the tandem repeats, as these are really good cytogenetic probes, giving very strong signals because it's the same sequence always next to each other. So you get really a really nice probe. So you can identify tandem repeats directly from the reads using different approaches. We used a read cluster approach, the Repeat Explorer pipeline. It's one approach that is used by a number of labs in the last time. So that's quite established, I would say. So you basically have the reads, they map against the monomers of the tandem repeat, and because this is all the same sequence here, they all cluster together and form clusters of a typical shape. So we collected all of these sequences and basically made fish probes from them and had a look. So we tried a lot, and in the end we found 92 positions on the chromosomes which we could mark, and opted in the end for a combination of 6 probes of 6 colors in fish, using 4 satellites and the 2 rDNAs, which giving us this image, for example, for crocus sativus here. And if you stretch the chromosomes and analyze them, you can see quite nicely what I want to show you, because you can see here, for example, chromosome triplet 7 and 8 has 3 times the same chromosome. It looks the same here. Then 1, 2, 3, and 4, they are all more or less the 2+1 configuration, but chromosome 5 is a bit different here. It has one very unusual chromosome here, and 2 which are more alike. It's a 1+1+1 configuration. So this is our starting point to basically get into it and compare and find out a bit more. So we had Crocus catawatianus and other possible progenitors, and we basically looked at them, performed the same analysis here. And this is just one example for Crocus pallasii, for example. So we performed the fish, had a look at the chromosomes. You can already see it's but also quite different here, for example, and tested other species. I don't go into the detail, just to give you an idea of what it looks like. So tested different species here and then compared. So we had here in red now the Crocus catawatianus, which is established widely known parent here, and we can see that it matches quite nicely. So everything that matches is now framed by black now. So 14 of the 24 chromosomes can be found actually in this Crocus catawatianus plant. And we assume that some rDNA sites can be lost, because this is actually also something that is quite well known that this can happen, that rDNA sites can get lost or may even move around on the chromosomes. So we don't really have to place that much of a focus on the rDNAs here. And all the other plants from other species tested here show much less matching chromosomes here. I don't go into detail here. There's one other chromosome coming, which we can find in Crocus oreocriticus, but all in all, it's much less here. So we kind of leave that out. But still, having a look at Crocus catawatianus, a few chromosomes, like for example, this chromosome 5 or this, we cannot explain. So there was the question how variable Crocus catawatianus is in itself. And when we ordered these plants, we received 5 corms. So from the 5 corms, one was actually wrong, one was something else. It had a different flower and it was basically wrongly assigned. So we were left with 4 individuals, and this just shows you one of the probes. And you can see from the 4 individuals we had, all had a different number of signals. So there was not one that was the same. So we can already say from that there is a huge variability inside in Crocus catawatianus as a species. So looking at it's a fish. I have the original images in my hidden slides. I don't put that out here right now here. I think you have seen a lot of that. So comparing this with the individuals here and some other accessions we received later on, we can say that variability inside Crocus cartwrightianus is probably enough to explain the variability in Crocus sativus. So we could find 21 out of the 24 chromosomes that we have in Crocus sativus by looking at a couple of different individuals. So we don't know if the original individuals, for example, here chromosome 5-1, here's my point like this one, we couldn't find. We don't know if that's still around there, but we are quite positive about that variation within the species is actually enough to explain the chromosomal configurations that we find in saffron crocus. So from our two initial hypotheses, we could scratch this one out, and we are left with this one here, only one parent. And we think that two cytotypes have been involved. So in cytogenetics, we speak about cytotypes, and that it's not a real allopolyploid. So we were, as you can imagine, discussing what is it called? Is it allopolyploidy? Is it polyploidy? And so on. In the end, it's an academic discussion. So I will tell you what we found here. So we have two cytotypes going into that, and we can say that Crocus cartwrightianus is very, very likely the parental species of the triploid. So you've seen that here. So we think that the two cytotypes went into it, probably with unreduced pollen on cytotype 1 and regular egg cells on cytotype 2, being combined and sterile, and yeah, basically by humans propagated and distributed all over the world. So we started here, and we can say that we know now here that Crocus sativus comes from Crocus cartwrightianus most likely, and we can say that the triploidization occurred by two cytotypes. So these are the two things we could say after that, and you can find our saffron story here. It's already published. But what I didn't tell you about today is that we had also one independent study, not by us, but by our partners, who had a look at the same thing by genotyping, by sequencing at Gatersleben. So if you have a look at our story here, you should also look at the story of our partners from the IPK. So we were actually a bit disappointed that it was not possible to publish it in the same journal and the same issue, but you know all how it is. So we tried a couple of times. So in the end, it's like this. Good. So for the acknowledgements, these are the people, this is Dresden again, by the way, but 100 years ago. So this is the Crocus team here, and this is a team from our new project where we will go a bit more into the epigenetics of Crocus, and will go very, very deep into sequencing, producing reference genomes, together with Frank Blattner from Gatersleben, who is a Crocus expert and taxonomist, and Björn Usadel. They're still written Aachen, now he's in Düsseldorf, so this is actually wrong, who is a specialist in sequencing. So we will go on working with saffron, and I hope you enjoyed it, and I'm open for questions if you have the time.
All right, thank you, Tony. And like last time, first of all, that was a wonderful talk, and I have a bunch of questions, but I'm going to let other folks start putting their questions in the chat box. As we did last time, that seemed to work really well, and let you address those directly. Okay.
Okay, that's good. I just have to close the chat. So,
okay. Yeah, and if anybody wants to also unmute their video or whatever while they're chatting with you during the exchange, I think that works pretty well, as long as it's in an orderly fashion, if you answering the questions. So.
Good. So I found the chat. So thank you for the for the nice things you are saying. Yes, morphological differences. I didn't really say much about that. So actually, crocus, we think that Crocus cartwrightianus was actually grown before triploidization happened. So people were already growing Crocus cartwrightianus at that time and were probably also selecting. And Crocus sativus, in comparison to Crocus cartwrightianus, has a more so the flower itself is more violet, but also the stigmas are more orange. They are longer, much longer, and probably has been selected phenotypically from that time. Yeah, that was the question for Robert. So I'm like,
let's see, there's quite a few there still. You've got.
Genau, yeah. I'm just
trying to scroll back.
Yeah. So I will first go to the tetraploids and hexaploids. So we haven't, so actually hexaploidy, we tried that a few times, and also in Gartersleben. In Gartersleben, they actually tried that for many years now to produce hexaploid crocus, obviously, just by doubling up the triploid. So far, it hasn't worked, but it doesn't mean that we're not continue trying. But in the end, with Crocus sativus, you're very fixed with the vegetation period. You can't really, so up to now, it hasn't really worked to shift it somehow. So you only have autumn to try, more or less. And then if it doesn't work, you wait a year and try the next year. So it takes a while. So far, it hasn't worked to double up for the hexaploids. But in crocus, there are actually also a few species, or there are different ploidy levels also there. Can you resynthesize a triploid? Yes, resynthesis is also something that is one of the things we would like to try. So far, it hasn't. Well, so far, some triploids have been produced, but they didn't have the properties yet. So it's really fine. Well, one approach would be to really find the correct Crocus cartwrightianus plant. So we have some really good ideas, especially in Gartersleben, where Frank, our partner, has a lab as well, and they really do the collection trips, going to Greece and collecting and trying there. So we are more on the cytogenetics molecular side, using the resources that Frank from Gartersleben acquires there, and they really go there and collect and trying to get this running. Okay, so if I look away, there's my second screen. I don't know. Is there cytological variation within Crocus sativus suggesting post whole genome duplication evolution? I don't know. I can't tell you. But good question. Yeah, good. Would be nice. Is Crocus cartwrightianus known to be parental for any other polyploids? I can also say not that I know of. Maybe. Also would be very good. Also good question. I never asked myself that. So yeah, good that it came up. And there was also, yes, if we can be certain that this was a single triploidization event. Of course not. We can not be super certain, but we can say that Crocus sativus is genetically similar in every accession that we tested so far. So we think it was just one event where it was propagated. We would imagine that there were some differences if there were two events. There are like two individual triploidization events where two crocus sativus lines were generated, which were cultivated. One would imagine that there were some differences, but we will probably also see in our new project where we will generate one reference genome, one for Crocus catwaltianus, one for Crocus sativus, and also collect different accessions and having a look into its DNA methylation there, if there is variation in there, because we know that the different accessions are adapted to different climates in the different countries, and if there is some variation there, we will have a look, and we will probably also see if there is indeed some genetic variation that we didn't see so far, hopefully. Okay. I know that I skipped one, but I don't find it right now.
It was a question about how old is the triploid? That was the one I think that got skipped.
Yes, how old is the triploid? So we can't date it correctly, but we think, so basically right now it's dated using the frescoes, and that there is some evidence there that saffron was cultivated, but we also know that Crocus catwaltianus was cultivated before saffron. So yeah, it's always a question if it's really saffron or is it the progenitor that was cultivated there. But most people use the 4,000 years estimate like for this. So it's still in evolutionary times really young.
Or we have time for maybe one more question before.
Okay, yes, I'm just reading because if I speak, I don't.
Oh, no problem. Just let me know.
Sorry, yeah, exactly. Was on that massive yellow block on chromosome 7. Is it biologic heterozygosity? Wait, I just go to the slides, right? To check. I go directly catwaltianus. Here for the rDNA. Yeah, it's heterozygous for the rDNA. Yes, that's correct, the one plant, but there are also other plants, like we have one individual where it's homozygous for the rDNA. We have some individuals which are heterozygous for the rDNA, and it's actually not so uncommon, not just Crocus catwaltianus, but we can also see this in other species. And actually, allium, onion, which is closely related to crocus, is one of the first plants where these so-called moving rDNAs have been described. So we think we don't really have to focus too much on the rDNAs for this. I think this. Transcriptomic variation among subgenomes. I don't know. We haven't looked. We will look at some point, probably. But so far, I can't tell you anything about the transcriptome, anything at all, actually. Yes.
All right. Well, I think that wraps up for this. That was wonderful, Tony. And thank you for a great talk. And I did not know that complex history of crocus was so interesting. And I look forward to seeing the next update on this next year.
Yes, great. So thank you for listening, for joining the talk, and for the opportunity. And I hand over.
All right. Yeah. And so up next, we get to change continents and clades of angiosperms here for Dr. James Beck, who's going to tell us about do giant goldenrod or Solidago gigantea cytotypes occupy different niches. James is currently a professor at Wichita State University, where he's been since 2011. Before that, he did his PhD with Barbara Shaw at Washington University. and was a postdoc at Duke with Kathleen Pryor and Mike Wyndham, as well as a postdoc at the University of Wisconsin, Milwaukee with Dr. Sarah Hoot. And with that, I'm going to let James take it away and tell us all about Solidago, and maybe it looks like a little buffalo grass as well. So.
Yeah. So just before I get going here, just wanted to clarify that the screen everyone's seeing is the one with the two pictures on it of buffalo grass and Solidago. Good. All right. Yeah, my title notwithstanding, I'm actually going to be talking about two species today. Both have large ranges in North America, and both are auto-polyploid series. And the goal here is to see if there's evidence that polyploidy has contributed to range size. I figure probably most people, participating in this, when someone shows you just a figure of a bunch of species ranges, it's at least a minimally interesting to you. I mean, the fact that some species have large ranges and others have small ranges is one of the oldest questions in ecology and evolutionary biology. And although dispersal limitations have something to do with it, the weight of the evidence appears to be that a lack of adaptability beyond range is probably most important. So you need to create new genotypes, particularly at the edge of your range, in order to expand your range. And autopolyploidy creates the opportunity to create dramatically different genotypes really quickly. And in these two species, we wanted to see if cytotypes tended to be found in different portions of the range, suggesting that maybe that's how these species had explored a large range. The idea that whole genome duplication has contributed to range size. All right, so we're going to get started with buffalo grass, Bucloidactyloides. This is a low-growing grass species that's one of the dominant species of North American shortgrass prairies. So these are the westernmost, the driest of the American prairies. Habitats that look like this. This is from Kansas. Buffalo grass is also important commercially as a low-growing, drought-resistant grass. It's become the kind of go-to native turf grass alternative, particularly in dry areas. And there's a growing industry behind buffalo grass. You can order a 3-pound bag of buffalo grass seed online and get it going in your lawn. It occupies this large range here. This is all, I plotted all the records I could find on public databases combined with our collections. For all practical purposes, it's most of the Great Plains of the US and a large adjacent section of northern Mexico. So 4 cytotypes are known, diploid, tetraploid, pentaploid, and hexaploid. We're not going to be thinking much about the pentaploid because we know that it's rare and it's also sterile. Our research was focused around 3 questions. Are all buffalo grass cytotypes equally common? Are they spatially random? And if they're not spatially random, do they tend to occur in areas with different abiotic environments? This was a master's thesis. My lab is master's only. So my student Jacob and I needed a plan that would allow us to get lots of samples in the course of 1 or 2 field seasons. So we did both traditional field work, but also aggressively incorporated herbarium sampling. Over 2 field seasons, we made over 220 collections, more or less 1 per site, but also obtained 357 herbarium samples during visits to 8 collections. Critically, a bunch of these were tied to chromosome counts. So for a lot of samples, we had tissue and also a matching chromosome count. We did some of these counts. We collected bud material and did meiotic chromosome counts for 27 of the individuals we collected. These are just traditional acetocarmine squashes like the ones that have been done since the 1920s. Here's a couple of them. On the left there, you see a plant with 30 pairs, so 60 chromosomes, a hexaploid from Kansas. And then on the right, you see 20 pairs or 40 chromosomes, a tetraploid from Colorado. So here are all of our counts. And I just should say quickly, I really love Tony's talk, and it's gratifying to see people doing cytology. But at any rate, if you want to do this kind of thing, just this week, our protocol for doing meiotic chromosome counts was published in Apps. There's a detailed print protocol as well as 2 accompanying YouTube videos that show you how to do counts like that. So just a little shameless plug there. So not only were some of our collections tied to chromosome counts, 266 of the 357 herbarium specimens were vouchers from chromosome counts done by John and Charlotte Reeder in the 1970s. And a huge thanks to the Rocky Mountain Herbarium for giving us an opportunity to look at these and sample from a lot of these. So we had a big set of specimens, and for many we already knew their chromosome number, but we needed to figure out the chromosome number of all of these things that weren't tied to previous counts. The answer is that we genotyped them and used those data to estimate chromosome number. First thing we had to do is make sure we could extract DNA from 364 samples, efficiently and economically. Those of you that know my work know that I commonly do these 96 well chromosome or 96 well DNA extractions. You put them in these big racks with ball bearings. You modify a reciprocating saw to crush the tissue. It powderizes the tissue. And then you get the nice satisfying boundary layer there when you add the isopropanol. If you have a bunch of herbarium specimens of various ages, you get a nice kaleidoscope of colors at that point. But the yield from this traditional CTAB is very, very high, typically 20 to 60 nanograms per microliter. And of course you get plenty of it. Another shameless plug, we just published a video and a print protocol for this 96 well extraction on YouTube as well. So any of you that want that, let me know. If you just Google 96 well CTAB extraction, you'll find it on YouTube. So for our molecular tool, we used a fairly simple traditional molecular tool, microsatellites. Microsatellites are kind of the Jeep of molecular tools. If you're going to a certain location, they'll get you there, and they're rugged tools, particularly with a lot of sheared old DNA. This is just quickly showing you that we could get all 14 loci from basically everything, regardless of age. So you can see we're going back 30, 40 years here, and we're still getting all 14 of our microsatellite loci. This is, of course, increasingly unsurprising. These days, people are getting all kinds of data out of herbarium tissue. As a quick aside, the mean age of our data set was 1986. I realize this is probably before some of you anyway listening to this were born, but if you were around, 86 is when the Statue of Liberty was reopened. The Space Shuttle Challenger disaster happened. Halley's Comet passed by Earth and was visible, and also the Chernobyl nuclear accident in what is now Ukraine. Anyway, so we're going back in time a little bit to get a lot of counts, or to estimate a lot of these. The estimation procedure is very straightforward. What you see here are alleles at 4 individuals. So at 4 individuals at a single locus. So in this first individual, this could be a diploid, a tetraploid, or a hexaploid, right? I mean, it could be a hexaploid with 6 alleles of 159 base pairs. However, the second and third individual couldn't be a diploid, right? There's too many alleles. And finally, the one at the bottom with 5 alleles could only be a hexaploid. So with these kind of data, if you type enough loci, eventually the ploidy of the sample in question will reveal itself. So as an example, here are the 14. Each row is an array of alleles at loci in this hexaploid. So a few of them, could be misleading. We might think it's a diploid, but then we see these and we realize, oh, it's got to be a tetraploid. But eventually you see loci that reveal that this thing is a hexaploid. It's got 5 and up to 6 alleles. And it turns out this approach really works well. We had 76 samples for which we had a count and a genotype, and this predicted cytotype 89% of the time. So there's a little bit of error in our data set, but it's pretty good. So then we went back to our samples. For the green ones, we just used the cytotype we knew from chromosome counts, but for the others, we used this genetic estimation procedure. And this is what we get. This is the map this is the cytogeography of Beckloidactyloides. First thing, our first question about rarity, both diploids and pentaploids are indeed rare. We really don't care about pentaploids, but since this is a crop now, we think it's important for people to probably go out and find and get some of this diploid tissue into germplasm collections for crop improvement. So we know that not all the cytotypes are equally common. Really, the story of this plant is tetraploid and hexaploid. And these 2 cytotypes are clearly non-randomly distributed. They're more or less on this northwest to southeast axis. For some context, these are the centroids of the 2 cytotypes. They're about 450 kilometers apart. And if you do a MANOVA on latitude and longitude together, these two cytotypes have statistically non-random distributions. So this is satisfying, right? I mean, we got a nice cyto map, you know, but these things are clearly geographically structured, but what does that mean ecologically? It certainly suggests that these cytotypes tend to be found in places that experience different climates. But let's do some formal statistical analyses using the bioclim variables. You're likely familiar with these. You have to get rid of some of them, you know, in order to get rid of you know, paired, highly correlated pairs of variables. So after we kind of pare the data set down, we're left with these 7 variables. And you can do an ordination. This is a plot. This is kind of ecospace, abiotic ecospace on principal component 1 and 2, which turn out to be basically temperature and precipitation. You can already just visibly see some ecological differentiation here. And if you do an ANOVA on those first 4 PCs individually, they differ at PC1, which was again temperature. And if you do a MANOVA on the first 4 PCs at once, they're definitely different. So just with kind of traditional statistical tools, these cytotypes do not occur in environments with the same abiotic conditions. We can also do a discriminant function analysis and ask which of those variables kind of informs the difference, explains variation between the 2 cytotypes the best. And the 2 highest scoring or highest loading on the discriminant function were features of the growing season, mean temperature of the warmest quarter, and precipitation of the warmest quarter. So it appears that the hexaploid has shifted to warmer, wetter growing season conditions, or at least it outcompetes the tetraploid there perhaps. We also did a niche equivalency test in ecospate, and these niches are not equivalent, although they overlap a lot. The hexaploid niche has expanded on the tetraploid niche by only 3%, so it's only explored kind of 3% new ecospace, and it's left only 2% of the tetraploid niche unfilled. So to answer our questions, all buffalo grass cytotypes are not equally common. They're not spatially random, and they do tend to occur in different abiotic environments, although this difference is subtle. That's how I would say it. It's a subtle environmental differentiation. All right, now to get on to the actual, kind of bait and switched you there, to get on to the actual, if any of you guys were goldenrod enthusiasts, we're finally going to get to this stuff. Like buffalo grass, Solidago gigantea has a large North American distribution. It's essentially, you know, everything east of the Rockies. It gets into southern Canada, so it's one of the most widespread goldenrod species. This, by the way, is my master's student Maria Martino's thesis, which she just defended 4 days ago. So congratulations to her. Similarly, we did some field work. This is to show you that these are giant goldenrods. We went out, we got tissue, some tissue that we could tie to chromosome counts. We documented the same 3 cytotypes that people have seen before: diploid, tetraploid, and hexaploid. Here's some of the new counts we did. Same strategy. We have a set of field and herbarium specimens, some of which are tied to chromosome counts. In this case, we had a giant set of chromosome counts supplied to us by our collaborator, John Semple, who's been studying these things for years. Our cytotype estimation procedure worked well again. Here's a diploid from Tennessee. You can see that all of the loci have either 1 or 2 alleles. Here's a nice hexaploid from Kansas. And so in black, you can see the loci that have 5 or 6 alleles. So again, same story. We used this estimation procedure. It worked about the same. It was about 85% correct. So again, there's a little bit of error here. Here's the distribution of our 629 samples. Remember, this is a mixture of previous counts, genetic estimation, and new counts. And again, there's this fairly obvious geographic structure. And since we had 3 common cytotypes, we did 2 different analyses. First, looking at the ecological shifts in the diploid to tetraploid transition, and then for the tetraploid to hexaploid transition. And we felt good about thinking about it this way, given the result of a nearest neighbor analysis on genetic distances. Diploids and hexaploids are rarely each other's nearest neighbor. So we thought that was fairly clear that there had been a diploid to tetraploid transition, and then later a tetraploid to hexaploid transition. So that's how we structured our comparisons. We again had to get rid of some highly correlated variables, so we ended up with I think 8 bioclimb variables. If we compare diploid and tetraploid, they're different. Again, they differ on 2 of the 4 first principal components, and they differ if you analyze all 4 of them simultaneously. The niches are not equivalent based on a niche equivalency test, but we still see kind of single digit niche expansion and unfilling. Tetraploid niche has expanded on the diploid niche by only 4% and left 6% of it unfilled. So a little more dramatic than buffalo grass, but still in what I would consider fairly modest niche expansion. Here are the 2 variables that loaded most highly on the discriminant function. But the bottom is precipitation seasonality. That's the ratio of the standard deviation of monthly total precipitation to just regular monthly precipitation. So it's just kind of a measure of how precipitation varies over the season. And then the top one is temperature annual range. So it's these variables of kind of shifts over the course of a month or a year that are really informing the difference between these 2 cytotypes. The hexaploid niche was very different from the tetraploid niche. It differed at 3 of the 4 PCs, and of course all 4 at once. The niches were not equivalent, and this time we saw a dramatic niche shift. The hexaploid niche expanded on the tetraploid niche by 27% and left 22% of the tetraploid niche unfilled. So we think this is probably the most interesting observation from these data, is that these two cytotype shifts in Solidago gigantea did not have kind of similar results. One of them, from tetraploid to hexaploid, was much, much more consequential ecologically. And here's the 2 variables that loaded most highly on the discriminant function. At the bottom, you can say precipitation seasonality really, really, really tells the story of the difference between these 2 species. Not species, these 2 cytotypes. So are all giant goldenrod cytotypes equally common? Yes. They are not spatially random. They're very spatially structured. And giant goldenrod cytotypes do occur in different abiotic environments, particularly the hexaploid. So just to finish up, we feel that this is going to be the case in a lot of these widespread autopolyploid species, that it appears that generating new cytotypes is one way that a species can explore a larger geographic range. At the top, you see the herbarium acronyms for all the collections that were kind enough to let us to sample from. A little funding there. Yeah, and I'll end with this. It's always more complicated, right? We don't know if any of these cytotypes are actually differentially adapted to these areas. And in reality, it would be nice to do common garden experiments, reciprocal transplant experiments to actually test for differential adaptation. But yeah, I'll leave it at that.
All right, thanks, James. That was a great talk. Like last time, the questions are coming in here on the chat window, so I'll let you handle answering those here while other folks put their questions in. Okay, and we've got plenty of time.
Okay, I see. Let's see. I see someone has asked if these cytotypes hybridize and if they have multiple origins. Hybridization is something that I can't address. I will say that we feel pretty confident that the higher cytotypes have arisen a bunch of times. We have this we have like a 14 locus data set, so we can look at just basic multivariate, scatter in geographic or in genetic space, or we can do UPGMA, stuff like this. And there's a ton of genetic diversity in the tetraploids and hexaploids in these things, which suggests that they're not just one successful clone. So I can say with confidence that these things are kind of recurrently being formed. Let's see. Robert Laporte asked if there's any indication that there's more uncertainty in ploidy inference in areas of contact or overlap. That's not something that we've looked at. I can say that that in general, when we get misdiagnoses, we tend to overdiagnose. So a tetraploid that we diagnosed as a hexaploid, a tetraploid that we diagnose, or a diploid that we diagnose as a tetraploid. So in other words, there's an extra allele floating around in there at some of these loci, which is, you know, either if you've scored microsatellites before, you know that sometimes there's a peak you have to decide whether you want to call or not. There's probably some machine learning that could be done here, feeding it these data and getting a better at not just what we do, which we just look for the maximum number of alleles at any locus. There's probably a better approach, a more informed approach with these data. Any mixed ploidy populations? We did look in buffalo grass. We did go into a few populations, and we found both. We found some populations that were fixed, or were all we could find were one cytotype or the other. However, we did find populations where there were two cytotypes. The kind of ratio of cytotypes, it was unsurprising. In other words, if you were kind of in tetraploid land and you looked at 10 individuals, you'd see 8 tetraploids and 2 hexaploids. So the kind of ratio of cytotypes you'd find within a population matched where you were in the range. But yes, we haven't done this in Solidago, but we definitely saw mixed populations in buffalo grass. So that's also evidence that these things are being recurrently formed. Okay, someone asked about morphological variation between the cytotypes. In Solidago, we actually looked at this fairly carefully. The diploids and hexaploids are pretty diagnosable. The diploids tend to have really hairy abaxial leaf surfaces. The hexaploids are almost always glabrous. The problem are the tetraploids. Not surprisingly, the tetraploids do a little bit of everything, which completely, of course, blurs the line between cytotype and morphology. We didn't do anything. We just literally looked at those two characters. We didn't do any kind of real morphometrics. My hunch is you're going to find that the extremes of the autopolyploid series differ, but that it's a continuous kind of thing. Someone asked about pentaploids. So what we did with pentaploids is we assumed that if we saw, if we maxed out at 5 alleles, we assumed that was a hexaploid. The reason we did is that, remember, we had like 250 chromosome counts from John Reeder in the buffalo grass example, and they found like 2 pentaploids. So we use that as an indication that pentaploids are extremely rare, and they sampled from Mexico all the way up into the Central Rockies. So that we excluded the possibility of estimating pentaploids with our data, although we did include some counted pentaploids there. John has occasionally, in goldenrods, John has occasionally observed triploids, and I mean occasionally meaning like twice in 350 counts. I don't think he's ever seen a pentaploid. So these things are being formed, unsurprisingly. The low level of them suggests that they're getting swamped out, you know, that they're not being formed enough where you're going to see them frequently with these data. Obviously they're sterile, so they're not going anywhere. They could reproduce vegetatively, but it's not enough to get them, it's not enough for them to be common. Someone asked, can you remind me, do higher ploidy levels occur in areas with more precipitation seasonality? So in let's see, I'm going to look that up. Yes, so in both cases there was a shift. In Solidago, in both cases there was a shift to more precipitation seasonality with the higher cytotype. So tetraploids occupied areas with more precipitation seasonality than diploids, hexaploids, to tetraploids as well. So yes. Could you let us know source of all the climatic variables? These were just the standard world climb, bioclimb variables. They're fairly easy. Even someone like me can download and play with these things in R. So these are just the standard Bio 1 through Bio 19. So it's fairly easy to extract data at presence and background locations. Could any of those extra peaks be real, i.e. aneuploidy? Absolutely. So there are limits to what we can do when we see an extra peak with a museum specimen. Obviously, we can't do flow and we can't do a chromosome count on that tissue. But yes, they could be. Wondering about aneuploidy and/or triploidy, pentaploidy at contact zones. We haven't looked into that. I guess what we can say is, particularly in both cases, a lot of these things have actually been counted and people have looked at the chromosomes, and there's not a lot of indication that we're seeing odd cytotypes or aneuploids at least in their counts. Not that they've intentionally sampled contact zones. All right, what do you think, Mike? How many more should I do?
At least hit mine there.
Where are you? Oh, yeah. Any evidence of differences in cytotype fitness in areas of unfilled versus filled niche spaces? We have done absolutely no performance with these things. So that would be the obvious next step with a study like this would be to see if these things actually outperform each other in areas where they're not found. So, you know, it's kind of interesting, but like even like 15 years later, the Ramsey study with Achalia is still kind of the gold standard, you know. So no, that's I need to partner with an actual ecologist. So if any of you out there are ecologists and don't mind growing goldenrods, let me know.
Yeah, that would be great.
And someone just noticed, even though it was a private message, someone pointed out that this estimation procedure can now be done with data that are more powerful than microsatellites. I mean, if you're set up, if you've got the money and the expertise, you might as well just get HIBE or RADseq data for these things. You can do the same estimation procedure with that data, and then you obviously have something that you're set up to do phylogenetics with. So microsatellites are great. They're kind of a nice gateway drug. They're good for students. The price point is a little lower initially, so they still work really well for this kind of study. But if you're set up to gather full genome or kind of subgenome data sets, you might as well just get SNPs or get HibeSeq data and do that. Oh, someone was foolish enough to say they're always up for growing plants. I have to write that down.
All right. Well, that's excellent. Well, hopefully this will get some connections, and maybe in another year we'll have or a year or two we'll have some answers on those issues. Well, I want to thank everyone again. Thank Tony and James for speaking to us today and giving us a great couple of talks here while we're all social distancing or not, depending on where you are now in the United States. And hopefully everyone stays safe and healthy. I will leave we still have another hour or so here of chat. So if people want to, or of a Zoom time. So if people want to hang out and talk a little bit, feel free. I'm going to stop the recording now, and that'll be uploaded later on this afternoon once it's finished processing. I'll post those links again to the website. And just say that next week or 2 weeks from now, we have a couple of other talks coming from Thomas Annenberg on neopolyploidy exacerbating nutritional limitations that affects resource capture and storage traits in Euchra, Solidago, and Zhang Li on ancient genome duplications and low rates of chromosome loss in ferns with high chromosome numbers. And so hopefully everyone will be able to tune in next week for a couple of different takes on polyploidy and plant genome evolution. With that, I'm going to